So a couple things before I get started. First, I wanted to acknowledge that the program doesn't list my Norwegian collaborators, but they were co-authors on this paper, were big parts of the project. I want to make sure they get some recognition. And second of all, if some of this sounds familiar, it's probably because I presented preliminary results from this paper at a previous HBES. The basic story is similar, but there's also a lot new to discuss, so it's my hope that there's plenty of interest for everyone here. So the topic of the day, oxytocin, a protein hormone that's produced in the hypothalamus, stored and secreted by the posterior pituitary glands. While oxytocin itself exists only in mammals, related signaling peptides such as mesotocin, vasotocin, and others exist across a wide range of animal taxa. And these tocin signaling peptides, as I'll call them, have an ancient evolutionary history, making their debut somewhere between 450 and 700 million years ago, around the time that corals and jellyfish split off from the rest of us in the phylogenetic tree. And among the diversity of roles for these signaling peptides, functions pertaining to reproduction appear to be some of the more ancient and conserved ones. To wit, a few years ago, a group of researchers discovered a new protein and set of receptors in the nematode C. elegans that closely resembled mesotocin, oxytocin, and the others, so they named it appropriately enough nematocin. But more interestingly, what they found was that by knocking out nematocin, either by blocking its receptors or preventing its production in some way, this interfered with the mechanics of their sexual functioning. So what this figure is showing, basically, to be blunt, the knockout nematodes couldn't figure out where to put it. So even in this organism, very distantly related to us, these tocin signaling peptides are pretty crucial for reproduction. And with the emergence of placental mammals, monotremes, and marsupials, oxytocin and mesotocin in a few species becomes important for lactation. But it's not just within lactation that the mother-infant relationship provides an important template for understanding oxytocin. Indeed, some scholars argue that the mother-infant dyad is the biological prototype from which we should understand its other social functions. I mentioned how it's crucial for lactation. It's also a hormone with important obstetric functions, such as inducing labor in pregnant women and preventing postpartum hemorrhaging. But most relevant for this talk are the behavioral correlates of oxytocin, which jointly pretty strongly suggest that oxytocin mediates the formation of the mother-infant bond via its activity during various types of mother-infant interactions, such as nursing, play, and responding to an infant's cries. So this gives us a starting point. Evolutionary biologists have suggested that oxytocin and related peptides have expanded from their original roles several times to perform new functions. Reproductive functions, as I mentioned, appear to be one of the more ancient roles. With the debut of mammals and monotremes, this oxytocin was co-opted to play a role in milk ejection and lactation. This was further co-opted to mediate mammalian maternal behavior, perhaps because of its pre-existing role in a maternal context. Finally, perhaps these nurturing and bonding behaviors, these functions rather, were extended and combined with its role in reproduction to mediate the formation of sexual pair bonds in certain species. Potentially it goes even further in its social functions, and that's been some more recent work. But while all these domains are important to understand for the full evolutionary story, most of the work I've done focuses on this domain, that of sexual pair bonding. So, why do we have this model of oxytocin expanding into the realm of sexual pair bonds? And here, seminal studies focused on voles, in particular the prairie vole. So prairie voles are monogamous. They form enduring sexual pair bonds that are characterized by a very strong partner preference, and it seems like this partner preference is formed from physical contact and copulation in particular. Sister species to voles, though, to the prairie voles, meadow and montane voles, are polygynous. They don't form enduring pair bonds. So based on this observation, scholars have used voles as a model through which to understand the biological forces behind monogamy. And in a really elegant series of experiments, vole researchers established several findings. Administration of oxytocin can lead to the formation of pair bonds even in the absence of mating. Oxytocin receptor antagonists can interfere with the formation of pair bonds even when mating does occur. And finally, oxytocin receptor density in several key brain nuclei differs substantially between individuals of monogamous and polygynous vole species. So a lot has happened between those vole studies 25 years ago and now, but I do think it's important to note that the human oxytocin literature, which is pretty famous, is fundamentally based on these studies from voles. So from this base, psychologists have examined the potential functions of oxytocin within human romantic bonds. And indeed, a number of findings are consistent with a positive role for oxytocin in bonding processes such as relationship problem solving, sexual responsiveness, as well as overall ratings of satisfaction and involvement. So, ask the average person what oxytocin is. Odds are decent, they'll say it's something like a love hormone or a trust hormone or a cuddle hormone. And as I think I've detailed, it's not like this belief is without support. This very large corpus of psychological studies, as well as a smaller sort of foundation of animal work, has helped create this picture of oxytocin as a biological basis for the psychological closeness and impact of diverse kinds of social bonds, whether it's the bond between mother and infant, between lovers, between people in a social in-group, or as this recent paper that got the cover of Science suggests, even the bond with your dog. And Peter Gray talked about that a couple days ago. So it sounds great, right? Well, unfortunately, I have to say not so fast. So put simply, there are a ton of controversies within oxytocin research. First, the literature isn't really all that consistent. So the substantial number of findings consistent with the notion of oxytocin as a generally pro-social hormone is contradicted by an almost equally large number of findings that implicate oxytocin in behaviors that would seem to be pretty detrimental to bonding, and I've listed some of them here. Second, from a methodological standpoint, measuring and manipulating the oxytocin system is pretty difficult. It's a large, water-soluble neuropeptide hormone, so it doesn't readily cross the blood-brain barrier, unlike smaller steroid hormones such as cortisol do. And it's become really popular to administer oxytocin to people through a nasal spray, but it's not clear whether this nasal administration is reaching the brain in any way. There are some small pilot studies indicating that potentially it can, but a route or mechanism still hasn't really been identified. It's a fragile, heat-sensitive hormone, so when you're taking samples from non-invasive media like saliva or urine, you often have to concentrate the sample to get a detectable signal, and this can be something that's very hard to do correctly, as I can attest to personally. And finally, oxytocin is released and circulates both within the peripheral circulation and the central nervous system. And almost universally, when we're talking about oxytocin research, we're talking about measurements taken from peripheral media. So we're implicitly assuming that what's going on in the periphery reflects what's going on in the brain, though again, this isn't necessarily the case. All right, as if that weren't enough, oxytocin research has also become one of the targets of scrutiny within this larger psychological debate surrounding reproducibility. So 2 recent reviews are relevant here. The top box comes from a meta-analysis on effects of oxytocin administration on trusting behavior. So this is really one of the first effects from this literature, probably considered by many to be sort of a foundational effect. Unfortunately, a meta-analysis of all the available studies was not able to find any mean effect, which is what that bottom diamond shows. The bottom one, this is from one lab that decided to open its file drawer, so to speak. They'd been doing oxytocin administration studies for over a decade, and they hadn't published a number of findings for any number of reasons. But what's really worrying is when they tried to open their file drawer, when they did open their file drawer, and they looked at all these effects that were just attempted replications of things that were already published, they basically found nothing, a bunch of null effects. So with all that said, can we make any sense of the literature? Can we extract a meaningful functional signal? Some don't think so, for some combination of the reasons I just mentioned. But I'm going to do my best to make the case that it can and should be done, all these important problems notwithstanding. A number of researchers have attempted to do this, to put forth an overarching conceptualization that makes sense of a confusing and complex literature. Our proposal, which is the one I've bolded at the bottom, isn't mutually exclusive with many of these others. But I do think, however, it diverges in some key ways, and I'll talk about them now. So our goal, rather than to examine effects of oxytocin administration, was to investigate what leads to the natural production of oxytocin. And we weren't the first to ask this question. Just within the domain of oxytocin and sexual pair bonding, there already existed 2 very prominent hypotheses regarding the link between natural oxytocin levels and romantic relationship quality. The first, named Calm and Connect, predicts a positive relationship between oxytocin and relationship quality. It's thought that this is because oxytocin inhibits behaviors that are detrimental to relationships like anxiety and defensiveness. This then sets the stage for future warm, nurturing interactions with the partner. So here the prediction is that involvement positively predicts an oxytocin release, and there's some support for this belief. The second idea is called tend and befriend, and this one predicts a negative link between relationship quality and oxytocin. Here, it's thought that distressful relationships trigger the release of oxytocin, with the function of this production to be to lead individuals to be motivated to seek social affiliation elsewhere, outside of the distressful bonds. And in support of this, Taylor et al. Find that participants who report more distress within the relationship have higher circulating levels of oxytocin. So I mentioned the oxytocin paradox. Here is seemingly one instance of it. But one thing I didn't mention from these studies is that involvement was measured via self-report, whereas distress was measured by having participants report on one's partner. So while these two findings are kind of painted as opposites often, it's entirely plausible that a relationship could have both, right? A highly invested partner that is still distressed because their partner isn't as involved as they are, or as involved as they would like at least. So jointly, this reflects what we have titled Identify and Invest, our cute little title to go along with the other two. So we believe that a discrepancy in relationship involvement leads to a perception of vulnerability within the relationship. And because one person highly values this relationship, this vulnerability triggers the release of oxytocin. And the function of this release is to orient allocation of psychological resources back toward the relationship. So not elsewhere, but back towards the bond that's in danger. So again, rather than involvement or distress triggering the release of oxytocin, we think it's one and the other simultaneously. So we tested these ideas in two different cultures, albeit both weird in the anthropological sense. The first was among college students at the University of New Mexico in Albuquerque. The second from participants in the beautiful city of Trondheim, Norway. So we recruited nearly 300 participants all told across the two studies. One sample from UNM had both members of the couple come in. The Norwegian sample had romantically involved individuals come in without their partner. So after informed consent, they completed an initial questionnaire mostly focused on their health, while simultaneously providing an initial saliva sample. After they were done with both those things, they went through our manipulation, which was a 10-minute thought listing task where we had them think about their relationship with their partner. You can see the prompt that we gave them here. After the task, they started on the second questionnaire, which asked them about detailed aspects of their relationship. So we performed a factor analysis on the overall battery of relationship questionnaire measures and extracted a number of factors. These constitute our predictors of interest here. And also, 15 minutes into the second questionnaire packet, they provided a second saliva sample. So 25 minutes elapsed between the two collection points, hopefully capturing an oxytocin response, which was our dependent variable of interest. We chose this 25-minute window based on some previous breastfeeding studies that were able to detect salivary oxytocin changes in this window. Okay, so here are the results from our combined analysis of both samples together in a mixed model regression. We included sex, study, and relationship length as covariates, non-significant, and relationship involvement, however, was a highly significant predictor going in opposite directions for self and for partner. So, as one's self-assessment of relationship involvement increases, so too does their oxytocin response. However, that same person's oxytocin response also increases as their partner's reported involvement decreases. And this effect was about the same between studies, about the same between men and women, and jointly these results support our hypothesis that high involvement and distress both are predicting an oxytocin response. So one way to simplify these results and present them in a single graph is to create a difference variable between self and partner involvement. And that's what we've done here. So statistically, this is just done by constraining the weights in the regression model to be equal in magnitude but opposite in sign. And so that's what the x-axis is here, sort of an involvement difference of self minus partner on a general factor of relationship involvement, and the z-scored oxytocin change on the y-axis. Again, as the difference in involvement increases, so too does the oxytocin change across the task. And this is true in multiple domains that emerged from the factor analysis, including two that we're calling love/bonding, and another one, sexual passion/responsiveness. So in sum, we found strong support for our predictions across two studies. Relationship involvement discrepancies appear to strongly predict an oxytocin response. So we're arguing that vulnerability might be a state that triggers oxytocin release. And one implication is that we think this framework could potentially fit for multiple classes of relationships. So for instance, mother-infant bonds and new romantic relationships are two other contexts that might be construed as vulnerable. Indeed, these are two other contexts in which people have consistently found elevated oxytocin levels. Perhaps there are other kinds of vulnerable bonds to investigate. One more immediate implication, again, is that This model yields predictions that can be readily